Our world is covered in plastic, and it's not fantastic. Most of the plastic we use ends up in landfills or natural environments where it can take many years to break down. But what if the plastic could sort of self-destruct? With the help of microbes, this is possible. Scientists recently produced a type of polyurethane, a commonly used plastic, containing spores of bacteria. When the spores germinate, the bacteria munch away at the plastic, causing it to disintegrate. This advancement is just one of many ways in which scientists are harnessing the metabolic powers of microbes to help solve some of the biggest challenges of our time. Welcome to Microbial Minutes, the American Society for Microbiology's update on what's hot in the microbial sciences. I'm Madeline Barron, Science Communication Specialist at ASM. Before we dive into today's video, please take a moment to fill out the survey linked in the description. It's 5 questions, takes less than 3 minutes, and your answers will help us make better videos. We appreciate the support. Now, we will get to the cool science, but first a fact that is decidedly uncool. Our planet is drowning in plastic. We produce 400 million pounds of the stuff every year, and less than 10% of that is recycled. This is in part because plastic is an umbrella term for a variety of products and formulations, some of which, for various material and infrastructural reasons, are more easily recycled than others. The result is loads of plastic floating around the environment and bulking up landfills. Now, when it comes to managing this plastic waste, microbes are emerging as key players. Some bacteria are metabolically equipped to degrade plastics within the environment, and this does happen naturally to an extent. However, the microbial degradation of plastic depends on conditions that support the growth of a given organism. Moreover, that particular organism must come in contact with the plastic it can metabolize, and this doesn't always happen. In a recent report, researchers leveraged microbes in a new way to address some of these concerns and add to what will be a multifaceted solution to our plastic problem. Their technology involves mixing spores of the widely used bacterium, Bacillus subtilis, into thermoplastic polyurethane, or TUP, a flexible plastic used in everything from footwear to electronics. Germination of the embedded spores gives rise to vegetative bacteria that eat away at the plastic matrix. Now, the key word here is spores. There have been many advancements toward incorporating live cells, including microbes, into materials to create hybrid engineering living materials. The reason why is living cells are self-replicating, self-regulating, and programmable in response to their environments. The downside is that cells are fragile. They require specific conditions to survive, including temperature and pH, and this is not always feasible in the context of commercial material production, where processes are characterized by deadly temperatures, chemicals, and more. Spores, however, are much hardier. These are metabolically dormant forms of bacteria that allow for survival in environmentally hostile conditions, like high temperatures, high pressure, toxic chemicals, and more. Like seeds of a plant, the spores contain the building blocks of mature vegetative bacterial cells, like DNA and enzymes. They can survive for years, germinating into cells within minutes when conditions are favorable, generally in the presence of nutrients. Here, the scientists generated strains of B. subtilis whose spores could withstand temperatures as high as 135 degrees Celsius for 30 minutes, the temperature needed for production and extrusion of TPU. Normally, B. subtilis can't last for more than a few minutes at 100 degrees Celsius, so this was a big step up. The strains were generated through a process called adaptive laboratory evolution, in which B. subtilis was exposed to progressively longer periods of heat stress, and the survivors, that is, those with mutations that allowed them to live under high temperatures, were isolated. With their heat-tolerant spores in hand, the scientists then mixed them with the TPU and extruded the plastic into long strips. And here's the notable bit. Once the scientists incubated the spore-containing TPU in compost, a medium similar to what might be in a landfill and has nutrients needed to trigger spore germination, it broke down faster than TPU without spores. This was true even if the compost had been autoclaved. The idea there was that microbial conditions are often not ideal to support plastic disintegration. By autoclaving, the scientists removed most viable microbes from the compost and could then determine how well the spore TPU breaks down, regardless of the environmental microbial community present. With that, after 5 months of incubation, there was a nearly 93% loss in mass of the TPU-containing spores, compared to roughly 44% of normal TPU. The researchers also found that adding spores actually improved the mechanical properties of the plastic itself, that is, increased its toughness, likely due to interactions between the spores and the plastic polymers. These findings indicate that spores embedded in the TPU could facilitate degradation of the plastic with minimal intervention and regardless of the other microbes present. They highlight the potential of using spores to create materials that are not only stronger, but come equipped with the tools for their own breakdown. Researchers point to scaling up production of the TPU from the lab to industrial scale as a key focus. In addition, TPU is just one of many different types of plastic and by far not the most difficult to degrade. This is evidenced by the fact that almost half of the regular TPU broke down in autoclaved compost after 5 months. The scientists chalk this up to the regrowth of degrader microbial strains in compost during incubation or non-microbial breakdown processes. The goal would be to expand this methodology and technology for application to diverse plastic products. It's also worth noting that this advancement does not represent the ultimate solution to plastic waste. Other scientists outside of this study highlight that products like this must be handled with care so that they don't foster the impression that plastic pollution is less of a concern because plastic can degrade more quickly. Again, depending on the plastic, even in the best of circumstances, it can take years and years for plastic to degrade. Ultimately, the rate at which plastic is added to the environment far outpaces how quickly it is broken down. Technologies like the one presented here represent one piece of a larger puzzle for how to deal with the problem, including reducing the number of plastic products produced and used in general, and recycling whenever possible. This work is also one example of an advancement that could propel the bioeconomy, the facet of the global economy that uses biological resources to produce goods and services across a diverse network of industries. Such technologies aim to reduce waste and, in some cases, repurpose it, and create sustainable solutions to some of our biggest challenges, including plastic waste, climate change, food security, and more. Within all of this, microbes are integral players. In fact, in addition to bioplastics, microbes are being harnessed to generate sustainable clothing, fuels, chemicals, and more. They are already central to food preservation and generation and will continue to be useful for everything from preserving bee populations to promoting the health of human populations. In this way, microbes and microbiologists hold the key to preserving and boosting the health of animals, humans, and the planet. That's all for today. Don't forget to fill out the survey mentioned up top. I'd like to thank you for listening, thank Ray Ortega for production, and I'll talk to you soon.